Non-Mendelian genetics refers to traits that are not controlled by one gene at one locus or by a simple dominant versus recessive system. For these traits, the expected patterns in the offspring are not observed. In fact, the patterns that are observed can't be explained by the simple predictions of Mendel's laws. And examples of traits that follow non-Mendelian genetics are polygenic traits, where multiple genes can control the characteristic; incomplete dominance, where the heterozygotes display phenotypes that are in between the dominant and the recessive phenotype; and examples of traits that are controlled through codominance, where both alleles in fact are dominant and fully expressed in heterozygotes. And there's also the case of sex-linked traits, genes that simply occur on the X or the Y chromosomes, and therefore their pattern of expression is predicted by the sex of the offspring. In polygenic traits, we have multiple genes that control a single characteristic, such as eye color. Eye color is a good example of a polygenic trait. In this case, there are multiple alleles, and they occur on different chromosomes. So there are alleles that lay down pigment for brown eyes, green eyes, and blue eyes. And these occur on at least two different chromosomes. Chromosome 15, for instance, has two alleles for eye color, the blue and the brown. And chromosome 19 has two alleles, one for the green and one for the blue. Now, eye color is determined by different amounts of melanin that's laid down in two different layers of the iris. And melanin is typically associated with a brown colored pigment. Now, in the back of the iris, the first layer, the brown colored melanin is laid down but appears blue because of the refraction and reflection of light rays off of this layer. The front layer of the iris will receive some varying amount of brown color, either darker or lighter pigment based on the gene that's controlling the eye color. And when these two layers are combined, one over the other, we get a certain eye color, such as gray eye color, as you can see with a lighter brown over the blue. In incomplete dominance, heterozygotes express a phenotype that's somewhere in between the dominant and the recessive phenotype. We call this an intermediate phenotype. And a good example of this pattern of inheritance is in the 4 o'clock flowers. 4 o'clock flowers can have a red color that's determined by being homozygous for the red gene, and a white color, which is homozygous for the white gene. But when we see the heterozygotes that are formed from, say, the mating of these two parents, their color is pink, somewhere in between the red and the white, or an intermediate phenotype. So in this case, there really is no dominant allele because there is no recessive allele. In this case, both of the alleles are expressed, and we get this kind of a blend, or an intermediate phenotype. Now, if we mated one of these two F1 offspring to heterozygous pink flowers, we will again see the homozygous red and the homozygous white individuals be formed, which would be red and white flowers as well. In codominance, both alleles are fully expressed in heterozygotes. So here we have two dominant alleles possible, two or more. And a good example of this pattern of inheritance is in the ABO blood group system. The inheritance of this depends on three different alleles. An A allele means that your red blood cells have an A antigen on their surface. You can see that here in this simple cartoon showing these A antigens on the surface of a red blood cell. Now, antigens are just molecules, sort of like little signals to the antibodies in your bloodstream that say, it's okay for me to be here, don't attack or don't mount an immune response. They're recognized by the antibodies in the plasma. Having the B allele means that your red blood cells are exposing the B antigen, just simple molecular markers on the surface of their red blood cells. And if you have the recessive lowercase I allele, that means that neither antigen is present on your red blood cell, and that's typical of a blood type of O. Now, to have these different blood types, for instance, blood type A, you can either be homozygous for the A allele or have the A allele and the lowercase I allele, in this case the recessive allele. Blood type B follows the same type of system. And then to be blood type O, you would have to be homozygous for the recessive allele. Okay, so the codominance part of this story arises when you have both an A allele and a B allele, because in this case both of these alleles are dominant. And here you would have a red blood cell that exposes both the A and the B antigens. In other words, both of the alleles are fully expressed and present. In this case, it's different from incomplete dominance, the last example with the 4 o'clock flowers, because the phenotype here is not some blend, or not intermediate between the dominant and the recessive phenotype. In fact, both of these traits are fully expressed and individually expressed at the same time. So, for instance, we see both the A and the B antigen on the red blood cell. Now, for sex-linked traits, we talk about that in the next video.